Dear viewers, greetings. Today we can discuss about types of rotors. These type of rotors mainly used in centrifuge apparatus for centrifugation process. Now we can know about centrifuge and also centrifugation process. Centrifuge is a device for separating particles from a solution based on the size, shape, viscosity of the medium. Based on this, we can separate it. Next, centrifugation is a process or the separation technique are based upon the behavior of particles in the applied electric field. And also centrifugation is one of the most important and also widely applied research technique in biochemistry, cellular and molecular biology and in medicine. Next move on to types of rotors. A centrifuge is an instrument which produces centrifugal force. The particles are normally suspended in a specific liquid medium held in tubes which are located in a rotor. Here we can separating the two types of centrifugation. One is low speed centrifugation, high speed centrifugation. Low speed centrifugation rotors are made up of brass, steel and also perspex. High speed centrifugation rotors are made up of titanium or aluminum alloy. Titanium alloy are most resistant to chemical corrosion. Next move on to there are different types of rotors, namely first one swinging bucket rotor, second one fixed angle rotors, third one vertical rotor, fourth one zonal rotor. Now we can briefly see it. First one, swinging bucket rotor. It has buckets that start off in a vertical position, but during acceleration the rotor swinging out to make horizontal position during the centrifugation. Initially we can kept in vertical position. During acceleration the rotors turned off for horizontal position during the acceleration. During the rotation, the particle streak against the wall of the tube and travel down the wall of tube's base, causing the disruption of zones of sample. This will be rectified. How means control of this disruption can be achieved by slowly we can accelerate it or slowly we can decelerate it. It is of use in density gradient centrifugation technique. The swinging bucket rotors especially used in density gradient centrifugation technique. This picture shows the first picture for as like we can kept in the sample during acceleration. Second and third tube like the sample will be separated. Particles move on like that. Finally, after stopped in the centrifugation, fourth tube like we can separate the sample. Next move on to fixed angle rotors. In fixed angle rotors, the tubes are located in holes in the rotor body set at fixed angle between 14 degree or and 40 degree. In between these angle we can kept it. It has proved valuable over the differential separation of particles whose sedimentation rate differ by significant order of magnitude. This is for fixed angle rotors. First tube like we can keeping the sample during acceleration, second, third the particles sedimented, finally we can get the fourth tube like sample separated. Next one, vertical tube rotors. A vertical tube rotors is type of centrifuge rotor where the sample tubes are held in a fixed vertical position, resulting in the shortest path length for particles and making it ideal for rapid high resolution separation like isoparsonic density gradients. These rotors are best for applications where speed is priority, such as nucleic acid and viral particles purification, but are not suitable for pelleting sample. It is especially used for nucleic acid viral particle separation, not for other pelleting samples. This is for vertical rotors picture. First tube like we can keeping the sample during the acceleration. The walls of tube particles moved, and then third tube like the sample will be separated. Finally, after the final stage, the sample fourth tube like we can got it. Next one, journal rotors. Yeah, journal rotors is a specialized centrifuge rotor designed for separating large sample volumes such as viruses, organelles, and macromolecules by using density gradient. This type of rotor is especially used for density gradient centrifugation technique. It has a large cylindrical chamber with radial partitions which allows for high resolution separation of particles based on the size, density, sedimentation rate. The journal rotors are often used for purification and isolation of large scale preparations and can be operated in either batch or continuous flow mode. This is for further type of journal rotor, batch type or continuous flow type. First we will see about batch type. The batch flow type is more extensively used than the continuous flow type. Compared to continuous flow, morely we can use it batch flow. There are also designed to minimize the wall effects that are encountered in swinging bucket and also fixed angle rotor. In fixed angle rotor, swinging bucket, there is some wall effects there. It will be rectified in batch type. Low speed batch rotors are designed to operate nearly 5000 rpm. High speed batch rotors are designed to operate up to 60,000 rpm. Batch flow type is used for separation and isolation of hormones, enzymes, macroglobulins, ribosomal subunits, viruses, and subcellular organelles from either plant or animal tissue homogenates. Next one, continuous flow type. The rotors are similar in shape to batch type journal rotor but differ in design because of continuous fluid flow in the rotor. They are designed for high speed separation or relatively small quantities of solid matter from large volumes of suspension. Useful for harvesting of cells and the large scale isolation of viruses. This is for schematic diagram for a batch flow rotor and also continuous flow rotors. Here first one for batch mode of operation, second one for continuous mode of operation. Here numbers mentioned for some parts. One for magnetic stirrer, 2 for EC cell, 3 for anode, 4 for cathode, 5 for DC power supply. This is for common to batch rotor and also continuous rotor. Sixth one reservoir, seventh one pump, eighth one effluent inlet, ninth one treated water outlet, tenth one settling tank, 11th one clear water out, 12th one sludge outlet, 13 recycling one. From the 10 settling tank, sample is settled. After that through the recycling process, again it will goes to 8th one effluent inlet. In this way continuously sample will be separated. Thank you viewers, thank you for your support.